Welcome to Lecture 8.3, in which we will go over cell cycle regulation and also relate it to cancer. Our learning objectives for this video are to describe the main checkpoints of the cell cycle, describe the role of cyclins and cyclin-dependent kinases, describe the role of growth factors in promoting cell division, explain the correlation between errors in the cell cycle and cancer, define proto-oncogenes, oncogenes, and tumor suppressor genes, and understand the role of p53 in the cell cycle and why it is often mutated in cancers. As we learned in the previous video, the cell cycle has many different phases. Throughout these phases, there are many controls in place to make sure that each phase is occurring in the correct order and that the next phase starts only when the previous phase has been successfully completed. There are three main checkpoints that we will discuss: the G1/S checkpoint that occurs at the end of G1, the G2/M checkpoint that occurs at the end of G2, and the M checkpoint or spindle checkpoint that occurs at the end of metaphase during mitosis. Each of these checkpoints have different things that are being checked. The G1/S checkpoint checks to see if growth factors are present, whether there are enough nutrients and energy available to go through with cell division, and whether the DNA is free of errors. If the answer is yes to all of these questions, the cell will continue to the S phase. Since this is when DNA starts being replicated and the replication cannot be undone, this marks an irreversible commitment to going through with the cell division. If the answer is no to any of these questions, the cell will not continue to the S phase and either wait for better circumstances or repair the DNA if there are errors. At the G2/M checkpoint, the chromosomes should be completely replicated. It checks that this is the case, and also that the DNA is free from errors. If the DNA is completely replicated and has no errors, the cell will continue to mitosis. If not, the cell will either finish replicating the DNA or repair the DNA. The M checkpoint or spindle checkpoint asks if the sister chromatids are all properly aligned along the metaphase plate and whether the spindles are properly attached. If the answer is yes, the cell will continue to anaphase and separate the sister chromatids, and eventually finish mitosis. If the answer is no, the cell will wait for the chromosomes to be properly aligned and also make sure that the spindles are attached properly. This ensures that there are no mistakes in the distribution of chromosomes between the two cells. Many of these checkpoints involve checking the DNA because some errors can lead to dangerous mutations. How does a cell cycle know what order it's supposed to go in? The answer is with proteins called cyclins and cyclin-dependent kinases. Cyclins are proteins named for how their concentrations have a consistent cycle throughout the cell cycle. This graph shows the concentration of different cyclins throughout the course of the cell cycle. Cyclin D slowly accumulates at the beginning and begins to degrade towards the end. Cyclin E is most active at the end of G1 and going into S, and plays an important role in the transition from G1 to S phase. Cyclin A accumulates and is at the peak concentration during G2 phase, while cyclin B is highest at the beginning of mitosis. Cyclins are ultimately regulators of cyclin-dependent kinases. These are kinases that are activated by cyclins, which is why they're cyclin-dependent. The cyclins help let the kinases know when to be active, and the kinases are the ones that actually push the cell cycle forward. They do this by phosphorylating different proteins, and the addition of the phosphate group acts like an on or off switch that activates or inhibits various proteins for the cell cycle. Growth factors are signals that bind to membrane receptors on the cell and tell it to divide. The membrane receptors they bind to are usually called receptor tyrosine kinases. You don't have to remember the name for this class, but just know that the signal from the growth factor leads to the production of G1 cyclins, which activate specific CDKs that bring the cell out of the G0 phase and into the G1 phase, eventually leading to cell division. Cancer is a disease that can develop when cell cycle progression goes wrong. There are many forms of cancer, but what they all have in common is that they begin with uncontrolled cell division. This image shows tissue containing normal cells on the left and cancer cells drawn in gray on the right. There is a big bulge and many more cells crowded into the region. Some of the cells are also breaking through this barrier so that they can travel to other parts of the body. The reason why uncontrolled cell division can lead to cancer is because with each cell cycle, DNA is being replicated, and mutations will naturally occur as a part of DNA replication. However, if the mutations are not caught and are genes involved in controlling the cell cycle, cell division becomes further uncontrolled such that more mutations begin to accumulate. The constant division of cells leads to the formation of the tumor, which is this bulge here, and this can eventually lead to cancer. Many of the genes involved in cancer development are genes involved in the cell cycle. Proto-oncogenes are normal genes that help promote cell division. For example, the membrane receptors that receive the growth factor signals promote cell division, and so do cyclins and cyclin-dependent kinases. These genes are important, like how the gas pedal is important for driving a car forward. The problem is if a mutation causes the proto-oncogenes to become overactive. This kind of mutation converts the proto-oncogenes into oncogenes, which promotes cancer by leading to uncontrolled cell division. This would be like if the gas pedal gets stuck and the car starts going too fast. Tumor suppressor genes are the opposite. They inhibit the cell cycle and are important for regulating and preventing uncontrolled cell division, like the brakes of a car. If the mutation causes them to stop functioning, this can also lead to uncontrolled cell division. It would be like if the brakes stopped working in the car and there is no way to slow it down. The HHMI has a nice interactive website for learning more about cell cycle and cancer. One important tumor suppressor gene is p53, sometimes referred to as a guardian of the genome. It is commonly mutated and not working in cancer cells. When p53 is working normally, it checks for damaged DNA in the G1 and G2 checkpoints and directs the cell to repair the DNA. If DNA repair ends up not being possible, p53 triggers apoptosis, which is the process of cell death. This prevents the cell with the mutation from passing the mutation on. However, when p53 is mutated, the cell continues to divide even though the DNA is damaged. Without the chance to repair the DNA or kill the cell divides into two cells with damaged DNA. Now the mutation will continue to get passed on, and with the cell cycle going continuously, more mutations may accumulate and lead to cancer. In this video, we learned that the cell cycle has multiple checkpoints, and the cell checks that there are sufficient nutrients in the environment, that the DNA is free of errors, and that the mitotic spindles are properly attached during mitosis. Cyclins are proteins that appear during specific points of the cell cycle, and they regulate cyclin-dependent kinases to drive the cell cycle forward. Growth factors signal to the cell to enter G1 and begin the cell cycle. Cancer is a disease that develops when there is uncontrolled cell division, which leads to the accumulation of mutations. These mutations can turn proto-oncogenes into overactive oncogenes, or make tumor suppressor genes less active. One of the most important tumor suppressors is p53, which prevents cells with DNA damage from dividing. In the next lecture, we will start discussing meiosis, which is how reproductive cells are formed. Thanks for watching.